Hi all, welcome to DOPA YouTube channel. Okay, today we are going to discussing about the chapter respiration in plants. The chapter dealing with many processes involved in respiration in plants like glycolysis, Krebs cycle, electron transport system, etc. Okay, let me discuss about the glycolysis of respiration in plants. Okay, glycolysis. Okay, do you heard about glycolysis? Glycose means glucose or sugar. Lysis means splitting. So simply we can say splitting of glucose is called glycolysis. Glycose means glucose or sugar, and lysis means splitting of glucose, okay, is called glycolysis. Splitting of glucose into two molecules of pyruvic acid, it is known as glycolysis. So during glycolysis one molecule of glucose splits into two molecule of pyruvic acid. It is simply called glycolysis. Glycolysis means glycose means sugar or glucose, lysis means splitting. We can call it simply splitting of glucose is called glycolysis. During glycolysis, one molecule of glucose splits into two molecule of pyruvic acid. What I say? One molecule of glucose splits into two molecule of pyruvic acid. This process occurs within the cytoplasm, or otherwise we can call it a cytosol. Simply the process glycolysis occurs in cytoplasm or cytosol. The process glycolysis occurs in cytoplasm or cytosol. During glycolysis, one molecule of glucose converted into two molecule of pyruvic acid. Okay, or otherwise we can say partial oxidation of one molecule of glucose into two molecule of pyruvic acid in the absence of oxygen. Okay, let me check the different processes occurs in glycolysis. I already said that one molecule of glucose converted into two molecule of pyruvic acid, that is called glycolysis. Okay, we know glucose is a 6-carbon compound, and this pathway respiratory substrate. What do you mean by respiratory substrate? What do you mean by respiratory substrate? Do you heard about respiratory substrate? Respiratory substrate are the compound that oxidized during respiration. What do you mean by respiratory substrate? The compound which are oxidized during the process of respiration is called a respiratory substrate. Example, carbohydrate, protein, fat, these are respiratory substrate. During glycolysis, respiratory substrate is glucose. During glycolysis, respiratory substrate is glucose, and it is a 6-carbon compound. Okay, then glucose converted into pyruvic acid. Pyruvic acid is a 3-carbon compound. Pyruvic acid is a 3-carbon compound, and glucose is a 6-carbon compound. During glycolysis, glucose converted into two molecule of pyruvic acid. Glucose is a 6-carbon compound, and pyruvic acid is a 3-carbon compound. So which is the respiratory substrate in glycolysis? Respiratory substrate in glycolysis is glucose. It is a 6-carbon compound. During glycolysis, 6-carbon compound glucose converted into 3-carbon compound, that is pyruvic acid. Okay, let me check one by one. So glucose, during the first step, glucose is converted into glucose 6-phosphate. Okay, how glucose is converted into glucose 6-phosphate? By adding one phosphate group, okay, in the form of ATP. What do you mean by ATP? ATP means adenosine triphosphate. It contains 3 phosphate group. Okay, ATP provide one phosphate group to glucose, then glucose converter into glucose 6-phosphate. Okay, totally there are 10 steps are there. It is an enzyme mediated process. Okay, partial oxidation of one molecule of glucose into 2 molecule of pyruvic acid through a series of enzyme catalyzed reaction. Okay, through a series of enzyme catalyzed reactions are called glycolysis. Once again, what do you mean glycolysis? Partial oxidation of glucose into 2 molecule of pyruvic acid through a series of enzyme catalyzed reaction. They are called glycolysis. Okay, before starting glycolysis, we know actually the product of photosynthesis or end product is sucrose, because glucose is synthesized and it will convert into sucrose. Then this sucrose again converted into 2 monosaccharides. Sucrose combined with water in the form of the enzyme invertase, and forming glucose plus fructose. Okay, glucose is an isomeric form of fructose. Before starting glycolysis, sucrose converted into 2 monosaccharides. Such as glucose and fructose in the presence of the enzyme invertase. Okay, before starting glycolysis, sucrose converted into 2 monosaccharides such as glucose and fructose in the presence of the enzyme invertase. Okay, then this glucose converted into glucose 6-phosphate. It is a first step. Glycolysis mainly consists of or completed in 10 steps. Okay, there are 10 steps are there. During the first step, glucose combined with ATP. That means ATP provide one phosphate group to glucose and it will convert into glucose 6-phosphate. Okay, in the presence of the enzyme hexokinase. Which enzyme catalyzes this process? Hexokinase. Okay. Sucrose converted into 2 monosaccharides such as glucose and fructose in the presence of enzyme invertase. Then glycolysis starts. During the first step, glucose combined with ATP. Okay, that means ATP provide one phosphate group to glucose and it will convert into glucose 6-phosphate in the presence of the enzyme hexokinase. During the second step, glucose 6-phosphate isomerized. We already said that glucose is an isomeric form of fructose. Then glucose isomerized to form fructose 6-phosphate. It is the second step of glycolysis. During the third step, again fructose 6-phosphate phosphorylated. What is phosphorylation? Addition of phosphate group into a compound is called phosphorylation. First step, when glucose converted into glucose 6-phosphate, phosphorylation takes place. Glucose phosphorylated to form glucose 6-phosphate. What is the meaning? Glucose addition of phosphate group to glucose. Okay, in the form of ATP. ATP provide one phosphate group, that is called phosphorylation. Glucose phosphorylated to form glucose 6-phosphate during the first step. Then during the third step, fructose 6-phosphate phosphorylated to form fructose 1,6-bisphosphate. During the first step and the third step, phosphorylation take place, addition of phosphate group. Okay, during the first step, glucose phosphorylated to form glucose 6-phosphate and that phosphate group provided by ATP. Same during the third step, fructose 6-phosphate phosphorylated to form fructose 1,6-bisphosphate. Here also phosphate group provided by ATP. ATP provide phosphate group to fructose 6-phosphate and it will convert into fructose 1,6-bisphosphate. These 4 compounds are 6-carbon compound. During the next step, fructose 1,6-bisphosphate split into 2-carbon compound, okay, or 3-carbon compound, 2 3-carbon compound. Fructose 1,6-bisphosphate is a 6-carbon compound. Fructose 1,6-bisphosphate. PA means phosphate. It is a 6-carbon compound and it split into 3-carbon compound, 2 3-carbon compound. First compound name G3P, that means glyceraldehyde 3-phosphate. G3P means You can see here glyceraldehyde 3-phosphate. What do you mean G3P? Glyceraldehyde 3-phosphate. It is a triose phosphate because it contains 3-phosphate group and 3 carbon atom. Okay, they are 3 carbon compound. Okay, they contain 3 carbon atom. Okay, glyceraldehyde 3-phosphate. We can call it simply as G3P or PGAL. G3P, otherwise called PGAL. What do you mean by PGAL? Phosphoglyceraldehyde. G3P means glyceraldehyde 3-phosphate. G3P means glyceraldehyde 3-phosphate. Okay, it is otherwise called PGAL. PGAL means phosphoglyceraldehyde. Okay, it is a 3 carbon compound. Next 3 carbon compound is dihydroxyacetone phosphate. Dihydroxyacetone phosphate. DHAP. What do you mean DHAP? Dihydroxyacetone phosphate. Once again I repeat, fructose 1,6-bisphosphate is a 6 carbon compound. It is split into 2 3 carbon compound. Okay, one is G3P and one is DHAP. Fructose 1,6-bisphosphate split into 2 3 carbon compound. First 3 carbon compound is glyceraldehyde 3-phosphate, or simply we can call it as G3P or PGAL, phosphoglyceraldehyde. It is a triose sugar, 3 carbon containing sugar. Another 3 carbon compound is DHAP, dihydroxyacetone phosphate. What do you mean DHAP? Dihydroxyacetone phosphate. Okay, tell me dihydroxyacetone phosphate. Both of them are triose phosphate. What do you mean triose phosphate? 3 carbon containing compound. 6 carbon compound, fructose 1,6-bisphosphate split into 2 3 carbon compound. 2 3 carbon compound such as glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate simply we can call it as G3P or PGAL. G3P means glyceraldehyde 3-phosphate. PGAL means same, phosphoglyceraldehyde. It is a triose sugar, 3 carbon containing sugar. Another carbon containing compound, 3 carbon containing compound is DHAP, dihydroxyacetone phosphate. Both of them are 3 carbon compound. From one fructose 1,6-bisphosphate, 1 molecule of G3P and 1 molecule of DHAP is formed. During the next step, DHAP, this compound is interconvertible. They are reversible compound. During the next step, DHAP, 1 molecule of G3P and 1 molecule of DHAP is formed. During the next step, 1 molecule of DHAP again isomerized to form 1 molecule of G3P or PGAL. Thus, totally 2 molecule of G3P or PGAL are formed. From fructose 1,6-bisphosphate, 1 molecule of G3P and 1 molecule of DHAP is formed. Okay, 1 molecule of G3P and 1 molecule of DHAP is formed. During the next step, DHAP again isomerized to form 1 molecule of G3P. Thus, totally how many molecule of G3P? 2 molecule. 1 molecule already present and it formed from fructose 1,6-bisphosphate. During the next step, DHAP again isomerized, DHAP or dihydroxyacetone phosphate again isomerized to form 1 molecule of extra 1 molecule of G3Ps are formed. Thus, totally 2 molecule of G3P are formed. Thus, totally 2 molecule of G3P are formed. 2 molecule of G3P are formed. Okay. Each G3P converter into 1,3-BPGA. Each G3P converter into 1,3-BPGA. This G3P and DHAP is called a triose phosphate because they contain 3 carbon atom, okay, and a phosphate group, okay. But it converter into 1,3-bisphosphoglyceric acid. 1,3-bisphosphoglyceric acid means BPGA. So each G3P, here G3P, 1 molecule of G3P and 1 molecule of DHAP is formed. Again DHAP or dihydroxyacetone phosphate isomerized to form once more G3P. Thus totally 2 molecule of G3Ps are formed. Each G3P converted into, listen carefully, 1,3-BPGA. 1,3-BPGA means 1,3-bisphosphoglyceric acid. What is the expansion of BPGA? Bisphosphoglyceric acid. Tell me, bisphosphoglyceric acid. Each G3P converted into 1,3-BPGA. 1,3-BPGA. Okay, you can see here glyceraldehyde 3-phosphate, totally 2 molecules of G3Ps are formed. Then each G3P converted into 1,3-BPGA. It's a triose bisphosphate. Okay, extra phosphate group is present here. Okay, at that time NAD plus reduced to form NADH plus H plus. During the first and third step, ATP will be consumed. Okay, you should notify the ATP consumption and ATP production. Where does ATP produced? Where does ATP consumed? First and third step. That means when glucose converter into glucose 6-phosphate, then fructose 6-phosphate converter into fructose 1,6-bisphosphate. That means first and third step. During first and third step, ATP will be produced or consumed. ATP will be consumed. No ATP production, ATP consumption occurs in first and third step. During the first and third step, ATP will be utilized. ATP provide one phosphate group. Okay, first to glucose, then it converted into glucose 6-phosphate. Then third to fructose 6-phosphate. Okay, then it converted into fructose 1,6-bisphosphate. Okay, once again, first and third step ATP utilization take place. Okay, during this step, G3P converter into 1,3-BPGA. 1,3-BPGA at that time, NAD plus reduced to form NADH. Okay, listen carefully. G3P converter into 1,3-BPGA. At that time, G3P provide, we know addition of hydrogen and addition of electron is called reduction or oxidation. Reduction. What is oxidation? Addition of oxygen, removal of hydrogen, removal of electron is called oxidation. Then what is reduction? Addition of hydrogen, addition of electron and removal of oxygen is called reduction. G3P provide two redox equivalents such as 2H plus and 2E minus to NAD plus. Okay, NAD plus is a coenzyme. NAD plus, okay, 2H plus and 2E minus provided by G3P or glyceraldehyde 3-phosphate donate 2 proton or 2 hydrogen and 2 electron to NAD plus. As a result, NAD plus undergo reduction. Addition of hydrogen and addition of electron is called addition of hydrogen and addition of electron is called reduction. That's why NAD plus reduced to form NAD plus reduce or reduction reduced to form. Tell me NADH plus H plus. Okay, how NADH plus H plus is formed? When NAD plus reduce to form NADH plus H plus. Reduction process require 2 proton and 2 electron. That means hydrogen and electron. This hydrogen and electron provided by G3P, glyceraldehyde 3-phosphate provide 2 hydrogen and 2 electron. That means 2 proton and 2 electron. That is redox equivalent to the NAD plus. As a result, NAD plus reduced to form NADH plus H plus. Okay, 1 NADH is equal to, do you know, 1 NADH plus H plus reduced to form 3 ATP, oxidized to form 3 ATP, not reduced, oxidized to form 3 ATP. Okay, NAD plus reduced to form NADH plus H plus. 1 NADH can produce 3 ATP. You simply just understand that respiration means energy production, energy synthesis, ATP production. 1 NADH can produce, finally 1 NADH can, after this process, 1 NADH can oxidize to form 3 ATP. Where does NADH produce? It may ask, where does NADH produce? When G3P, when glyceraldehyde 3-phosphate converter into 1,3-BPGA or 1,3-bisphosphoglyceric acid, NAD plus reduced to form NADH plus H plus, and finally 1 NADH can produce 3 ATP. Okay, how this NAD plus reduced? Reduction process require 2 proton and 2 electron. That 2 proton and 2 electrons provided by glyceraldehyde 3-phosphate. Okay, glyceraldehyde 3-phosphate provide 2 proton and 2 electron to NAD plus and forming NADH plus H plus. NADH plus H plus. And finally 1 NADH oxidized to form 3 ATP. Okay, so NADH produced here. When glyceraldehyde 3-phosphate converter into 1,3-bisphosphoglyceric acid or BPGA, NAD plus reduced to form NADH plus H plus. When glyceraldehyde 3-phosphate converter into 1,3-bisphosphoglyceric acid, when glyceraldehyde 3-phosphate converter into 1,3-bisphosphoglyceric acid or 1,3-BPGA, NAD plus reduced to form NADH plus H plus. Okay, 1 NADH can oxidize to form 3 ATP. Glyceraldehyde 3-phosphate provide 2 proton and 2 electron. That means reduction equivalent or reducing equivalent to or redox equivalent to NAD plus. As a result, NAD plus reduced to form NADH plus H plus, and 1,3-bisphosphoglyceric acid is formed. Okay, then 1,3-bisphosphoglyceric acid converter into 3-phosphoglyceric acid. Okay, do you remember or recall the 3-PGA? 3-PGA we studied in photosynthesis Calvin cycle, first stable product. Okay, 1,3-BPGA converter into 3-phosphoglyceric acid. At that time, ATP is produced. Okay, don't forget to notify the or listen the ATP production and ATP consumption. First and third step, during first and third step, ATP will be utilized. Okay, during this step, when G3P converter into 1,3-BPGA, NADH is produced. During the next step, 1,3-BPGA converter into 3-PGA. At that time, 1,3-BPGA converted into 3-PGA. At that step, ATP production take place. ATP production take place. 1,3-BPGA provide 1 phosphate group to ADP. Okay, listen carefully. 1,3-BPGA or 1,3-bisphosphoglyceric acid provide 1 phosphate group to ADP and forming 3-PGA plus ATP. 3-PGA plus ATP will be produced. It is the first ATP production in glycolysis. 1,3-bisphosphoglyceric acid is substrate. Okay, in this reaction, 1,3-BPGA is called substrate. Substrate provide 1 phosphate group to ADP. 1,3-BPGA provide 1 phosphate group to ADP. As a result, it form ATP. Adenosine diphosphate means 2 phosphate groups are present, added with 1 phosphate group and forming adenosine triphosphate. Okay, this kind of ATP synthesis is called the substrate level phosphorylation. This kind of ATP synthesis is called the substrate level phosphorylation or substrate level ATP synthesis. Phosphorylation means synthesis of ATP. Substrate level phosphorylation. What do you mean by substrate level phosphorylation? Synthesis of ATP. Then phosphate group provided by substrate. In this reaction, substrate is 1,3-BPGA. In this reaction, substrate is 1,3-BPGA. That 1,3-BPGA provide 1 phosphate group to ADP. As a result, it form ATP. This kind of ATP synthesis is called a substrate level phosphorylation. Substrate provide 1 phosphate group. As a result, ATP will be synthesized. This kind of ATP synthesis is called the substrate level phosphorylation. So, in this reaction, 1,3-BPGA converter into 3-PGA, substrate level phosphorylation take place. Here is the substrate level phosphorylation. Substrate level phosphorylation. Okay, 1,3-BPGA provide 1 phosphate group to ADP. As a result, ATP will be produced. During the first and third step, ATP utilized. This step, 7th step, ATP production take place. When 1,3-BPGA converter into 3-PGA, ATP will be produced. How? 1,3-BPGA provide 1 phosphate group to ADP. As a result, ATP will be synthesized. This kind of ATP synthesis is called substrate level phosphorylation. Substrate 1,3-BPGA provide 1 phosphate group to ADP. As a result, ATP will be synthesized. Okay, and product is 3-PGA. Then 3-PGA converter into 2-PGA. Phosphoglycerate means phosphoglyceric acid. Okay, pyruvate means pyruvic acid. 8 means acid. Okay, 3-PGA converter into 2-phosphoglycerate or 2-phosphoglyceric acid. Simply we can call it as PGA. 2-PGA. Okay, 3-PGA converter into 2-PGA. Then 2-PGA converter into 2-phosphoenol pyruvate. That means 2-phosphoenol pyruvic acid, PEP. Okay, PGA converter into phosphoenol pyruvate or phosphoenol pyruvic acid. That is 2-PEP. Okay, 2-phosphoglycerate or 2-phosphoglyceric acid converter into 2-phosphoenol pyruvate. 2-phosphoglyceric acid converter into 2-phosphoenol pyruvate or 2-phosphoenol pyruvic acid. Okay, at that time 1 molecule of water will be removed. 1 molecule of water will be removed. And the last step, 2-PEP converter into 2-pyruvic acid, phosphoenol pyruvic acid or phosphoenol pyruvate converter into pyruvic acid. That is the 10th step of glycolysis. That is the 10th step of glycolysis. During the 10th step, same process, substrate level phosphorylation take place. Here also, substrate level phosphorylation or substrate level ATP synthesis take place. You already discussed about substrate level phosphorylation. Here, during this step, 1,3-BPGA is substrate and they provide phosphate group to ADP and forming ATP. Then same step, 10th step, 2-PEP is the substrate. 2-PEP provide 1 phosphate group to ADP and forming 2 molecule of pyruvic acid. PA means pyruvic acid plus ATP. Okay, same process. We already discussed about substrate level phosphorylation. Same process repeated here, but substrate is changed. Okay, during this reaction, substrate is 1,3-BPGA and this reaction, substrate is 1-PEP. PEP provide 1 phosphate group to ADP. Phosphoenol pyruvic acid or phosphoenol pyruvate provide 1 phosphate group. Okay, substrate is phosphoenol pyruvic acid or PEP. PEP is the substrate and that substrate provide 1 phosphate group to ADP. As a result, 2 molecule of pyruvic acid and ATP will be formed. This kind of ATP synthesis is called substrate level phosphorylation. Substrate level phosphorylation. So, during glycolysis process, in 2 places, substrate level phosphorylation take place. 7th step and 10th step. Okay, 7th step and 10th step, ATP synthesis take place. Okay, it may ask, where does ATP synthesis take place? 7th step and 10th step. Okay, accurately we can call it as when 1,3-BPGA converter into 3-PGA, ATP is produced. Then last step, when PEP converter into pyruvic acid, phosphoenol pyruvic acid converter into pyruvic acid also, ATP produced. These 2 kind of ATP synthesis in glycolysis is called substrate level phosphorylation. Substrate provide 1 phosphate group to ADP and forming ATP. Okay. When 2-PEP converter into pyruvic acid, ATP will be produced. ATP will be produced. Okay. Here also, when 1,3-BPGA converter into 3-PGA, ATP will be produced. Okay. In 2 places, ATP is synthesized and 2 places ATP is utilized and 1 places NADH is produced. Okay. Total gain of ATP, how many ATP produced during glycolysis? Okay. Each G3P, here you can see 2 G3Ps are formed. Okay, totally 2 glyceraldehyde 3-phosphate are formed. Each G3P converter into 1,3-BPGA. So first reaction, 1 NADH is formed. Okay, from each G3P, 1 NADH is formed. Totally 2 molecule of G3Ps are formed. So 2 G3P can produce 2 NADH. Okay, 1 NADH is equal to 1 NADH plus oxidized to form 1 NADH oxidized to form 3 ATP. Okay, each G3P can produce 1 NADH. So totally 2 G3Ps are formed. So 2 G3P produce 2 NADH. So 2 NADH can produce 2 into 3 ATP. That means 6 ATPs are the contribution from NADH. Okay. Totally 2 G3Ps are formed here. In this step, totally 2 G3Ps are formed. 2 G3Ps are formed. Okay. Glyceraldehyde 3-phosphate, totally 2 molecules are formed. Each G3P can produce 1 NADH. Thus, totally 2 G3Ps are there. So, 2 into 1. 2 into NADH means totally 2 NADH are formed. 2 NADH are formed. Okay. 1 NADH can produce 3 ATP. So, 2 NADH can produce 6 ATP. 6 ATPs are formed. 6 ATPs are formed from NADH. Then same. Here 1 ATP is 1 molecule of ATP. Here also 1 molecule of ATP from 1 G3P. So totally 2 G3P, 2 into 2, 4 ATPs are formed. Okay. Totally 4 ATPs are the formed. 4 ATP are formed. 6 plus 4, 6 ATP plus 4 ATP equal to 10 ATP is the total production of glycolysis, but 2 ATP consumed first and second process. During first and third process, during first and third step, 2 molecule of ATP is utilized. Okay, then we minus that 2 ATP, 10 minus 2 equal to 8 ATP is the net gain. Important question. How many ATPs are produced? 8 ATPs are produced, okay, during one glycolysis. Totally 8 ATPs are the net gain. Okay, how this 8 ATP formed? Totally 1 NADH is formed from 1 G3P. Totally 2 G3Ps are formed during glycolysis. So 2 G3P can produce 2 NADH. 1 NADH is equal to 3 ATP. That's why 2 NADH is equal to 6 ATP. 6 ATP is the contribution from 2 NADH. Okay. Then totally 2 molecule of ATP formed from each G3P. So 2 G3P can produce 4 ATP. 6 plus 4 is equal to 6 plus 4 is equal to 10 ATP. 10 ATP is the total production, and 2 ATP consumed during first and third step. First and third step ATP will be utilized. That 2 ATP will be minus. So 10 minus 2 is equal to 8 ATP. 8 ATP is the net gain of glycolysis. Don't forget, 8 ATP is the net gain of glycolysis. This is about glycolysis. Then what are the product of glycolysis? During glycolysis, 1 molecule of glucose converted into 2 molecule of pyruvic acid plus 8 ATP. This is the net gain of ATP. Okay. Okay. During glycolysis, 1 molecule of glucose converted into 2 molecule of PA means pyruvic acid. 2 PA means pyruvic acid. 2 pyruvic acid plus 8 ATP. Okay. During glycolysis, 1 molecule of glucose converted into 2 molecule of pyruvic acid and 8 molecule of ATP. This is the net reaction of glycolysis. During glycolysis, 1 molecule of glucose partially oxidized to form 2 molecule of pyruvic acid plus 8 ATP through a series of enzyme catalyzed reaction, and the net products are 2 molecule of pyruvic acid and 8 ATP. Net gain of glycolysis is 8 ATP, and key product of glycolysis is pyruvic acid, 2 molecule of pyruvic acid. Very important topic of this chapter, respiration plants. Don't forget the diagram and net gain of ATP and the net reaction. 1 molecule of glucose converted into 2 molecule of pyruvic acid plus 8 ATP. Okay. Okay. Bye.